So what is the genome? It's a person's complete set of genes, what makes us who we are and how we look. But why do we care? This is the sole explanation for human diversity, why we're all different from one another. But current research shows that there may be similarities between your genome and your great-great-grandfather's genome. Then what is the Human Genome Project? It was a huge breakthrough in DNA research which allows us to determine the sequence of all the nucleotide base pairs, giving us a complete map of the human genome. This laid the groundwork for further research to be done into the diversity of the human race.
What is the Simons Genome Diversity Project? A current project being performed by scientists takes a deeper look into the genome. They are taking 300 individuals from 142 populations in order to observe variations in genetics, linguistics, and cultures. They have developed new softwares with greater capabilities in order to bridge the gaps within the 1000 Genome Project, allowing them to map even more difficult genomes. How exactly is diversity measured? In this specific project, the heterozygosity of samples was measured. Heterozygosity is a measure of diversity because when a pair of alleles is heterozygous, each allele is different. Therefore, when looking at simple Mendelian Punnett squares, it is evident that there is a greater chance that offspring will have a different combination of alleles from parents or siblings. Where can we find this diversity? All over the earth, but the most diversity is found in sub-Saharan Africa. An example of this diversity lies within the African pygmy hunter-gatherers. The African pygmy hunter-gatherers have reduced X to autosomal diversity ratios in comparison to other sub-Saharan African people groups. There is an expected X to autosomal ratio that is constant among humans. When the ratio deviates from the norm, it is due to changes in demographics, including male-to-female ratios and overall population size. Natural selection also plays a role in the changes in the X to autosomal diversity ratio changes. In the Simons Genome Diversity Project, DNA samples are taken from populations that lived hundreds to thousands of years ago and compared to later samples of DNA.
About 200,000 years ago, the human population on Earth began to develop substructures. In other words, people groups began to separate and became diverse in location, culture, and in their DNA. Researchers working on the Simons Genome Diversity Project have been able to create a phylogenetic tree that traces the substructures of human populations, showing a timeline of when people groups began to show diversity in their genomes. The human species is compared to chimpanzees because chimpanzees are an outgroup for the human species. So how did we become different? Part of the research done by these scientists looked into the rate of mutation within populations. Mutations within populations' DNA sequences occur at a constant rate. Therefore, this is why we are able to use DNA sequencing and rates of mutations to understand the time that has passed between DNA samples. If the mutation rates among different people groups change, it shows the people groups are becoming more diverse. Also, the greater the variation in the mutation rates, the more time has passed between the lifetimes of the two groups sampled. However, they found a slight excess of mutations within non-Africans in relation to the sub-Saharan Africans. They were able to ensure the unbiasing of this experiment by mapping all the way back to the chimpanzee and only analyzing the X chromosome of males. While the Simons Genome Diversity Project does not have enough scientific data to prove this significantly, they have discovered trends that show promising results for the genetic changes that have had meaningful contribution in the human genome within the last 50,000 years. It is evident that the human species has had significant environmental, cultural, and behavioral changes that have rapidly transformed selection pressures and therefore have had great influence on our genomes.